Hi, my name is Gigi Maduakor. I am an upcoming senior bio major and chemistry minor at the illustrious Howard University. And this year, I got to work with NIH's HuBMAP program and Pacific Northwest National Laboratory on a project that is the optimization of same single cell spatial proteomics and transcriptomics. I'd like to say that I'm going to continue working in PNNL until August 22nd. So there's still some work to be done on this project, but for now, I would love to present to you what I have worked on. An overview of the NIH HuBMAP collaboration with PNNL. There is what HuBMAP is trying to do is that they're trying to map the human organs and provide a database that will be publicly available for people to use and view. There's an interest they have in the organization of biological processes, and there are several labs that are working to categorize and catalog proteins and peptides within the organs. The PNNL involvement is that they're focusing on pancreas mapping, and throughout our project, they are applying and developing new technologies for protein mapping at the single cell resolution. So for our project, we are trying to do parallel single cell proteomics and transcriptomics, which is the nanosplits project, which I'll explain more about right now. My specific involvement on the project is optimizing the nanosplits technology. First, I want to talk about the pancreas tissue, just so that you guys understand what's going on. The function of the pancreas is a lot of sugar and metabolic regulation processes. In the organization of the pancreas, there's a lot of different um, cell types, but there is specific cells such as the actin and the islets. Um, the islets themselves is something that we are focusing on because within the islets there are a lot of different cell types, but the four major ones, beta, alpha, delta, gamma cells, we're trying to catalog and categorize. So for our project, in order for us to interpret the spatial organization of the islet cells, we are going to be using mass spectrometry for protein mapping. Sorry, we are using mass spectrometry for protein mapping, and we're looking at things on a single cell level, which is higher than the resolution of an islet. It's really, really small. So there's a lot of challenges with that. And here's how we approach these challenges. Discussing mass spectrometry and its involvement in this project, PNNL is working on a focus of top-down proteomics, but my specific area of interest is bottom-up. And the difference between the two of these is top-down proteomics is a focus on the intact proteins, full size, but the bottom-up approach is where we have divided up and cut up these proteins and peptides. And once we get the data analysis from them, we are reconstructing them to interpret which proteins and peptides are actually in there. Mass spectrometry, if you didn't know, is a way to examine the structure of molecules by detecting the mass to charge ratio. Um, and what that means is basically you're going to get you guys can't see my mouse, sorry. Um, you're going to get a kind of array right here, which will tell you the mass to charge, and different sizes correspond to different proteins. And in order for us to get the bottom-up approach, we actually wind up separating the peptides and proteins beforehand using liquid chromatography and put it through the mass spectrometer, which will detect the ions. So the separation step is actually, I actually found out was a very long step. So there's a lot of work that went into that step, and I will elaborate on it right now. So within this project, NanoSplits uses a technique called nanopots, which was developed within PNNL. Um, and nanopots, if I click here, here's a video that plays that shows nanopots. It has these specific nanopot chips, which are shown on the video right now. They are very small and they contain nanowells. These nanowell chips are nanopots chips are very specially designed because they have these small, tiny little wells. that have nanoliter sizes, and they have hydrophilic and hydrophobic surfaces so that the dispensing cannot exit or enter the wells. These wells are designed to create less surface area, which cause less protein loss. The dropless positions are maintained so that despite all the moving around that you have to do for nano splits, none of the samples are lost, and the small nanoliter volumes increase reaction efficiency for certain parts of the uh, project. So, how do we use these nanopatch chips? After the creation of these nanopatch chips, we have to fill them. So, to fill these nanopatch chips, we utilize two different devices. There is the nanopatch loading robot, and then there's the Cell-in-One machine. And the differences between these machines, they're very similar. They fill in a small liters, but they have different purposes. For example, the nanopots loading robot specifically only fills in nanoliter sizes, and it involves a syringe, which makes contacts with the wells, unlike the Cell-in-One, which is able to fill even up to picoliter sizes, which is very specific, um, and utilizes piezoelectric dispensing, which means that there's the syringe on the Cell-in-One is not actually making contact with the chip, unlike nanopots. And also, Cell-in-One is capable of single cell sorting. So the Cell-in-One is actually commercially available, whereas the nanopots loading robot was designed within PNNL. So if you're any lab that is using the Cell-in-One, it's actually probably something you've heard of before, or you can comprehend what its uses may be. So after we filled the nanopatch chips, how do we capture spatial information? So the chips are filled with a buffer using the nanopots loading robot or the Cell-in-One. But now we actually have to put our samples inside it. And as we said earlier, we're looking at single cells within the islets, which is extremely small. So what we use is a machine called the LCM microscope or the laser capture microdissection microscope. And here in the picture to the right, you're able to see how the setup is. We'll have our tissue slide with the pancreatic tissue, which is already stained and prepared. Um, and you'll put it on top of the dissection microscope. And above that, you'll have the nanopatch chips, which is filled with the buffer. In the image, it's DMSO, but we're currently using a specific type of buffer, which I'll get into later. And in the nanopatch chips, the laser will actually cut out the shape, which is shown in the picture on the left, and it will catapult um, the sample into the nanopatch chips. So you'll have a ton of micro-sized wells with these little square sample sizes. Oh, and here's a video of how it cuts it out. And then it disappears. That's when it catapults. So now that we have it in the nanopots chips filled and we have the samples inside, this is the portion that is the nano splits, which is how we're going to be doing parallel transcriptomics and proteomics. I want to briefly explain why we're trying to do parallel transcriptomics and proteomics. Imagine that you have a cell, and a single cell is extremely small. It's really difficult to perform two different types of data analysis on it. But we want to be able to do two different things on it, but we can't do it with the cell next to it because what if the chemical makeup of the cells aren't the same? So what nanosplits aims to do is to split up the cell into two halves and amplify those cells with PCR so that we're able to perform transcriptomics and proteomics on the same cell. So how do we do this? We have the nanopatch chips, and we have to perform an extraction step that is designed to split the protein and RNA into two separate chips. Um, the portion of the lab that I'm focused on is optimizing these splitting techniques because initially they were seeing some 90-10 splitting and we're trying to get it 50-50. So the diffusion of the protein and RNA into the cells is non-specific. So you'll get some protein and some RNA in both chips. You'll take the nanopots chips, which has the sample size in it. Then you'll have another nanopots chips, which only has the buffer and no sample, and that'll be your acceptor chip. Then you'll layer the donor and the acceptor chip on top of each other, and which the cell, DNA, RNA, and protein will split up between the two chips. Then we'll use PCR to amplify what is it within these wells, and through that we can perform proteomics and transcriptomics and mass spectrometry in order to analyze the effectiveness of the splits. Um, so this is the portion that is the nanosplits portion. So for the nanosplits extraction optimization, our buffer is actually a citrate buffer because one problem with trying to work with RNA and protein is that, especially at these extremely small sizes, RNA is very, very fragile. There was challenges in not having the RNA degrade or deteriorate. Um, so we needed to design a method that satisfies both RNA and protein molecules. So here's the specifics of our buffer, which reduces mRNA hydrolysis and stabilizes the RNA. The reducing agent helps denature RNA, and the DDM reduces nonspecific absorption of protein and increases the low salt increase the reaction efficiency and stopped RNA and DNA from binding to our glass slides. So here's our first run of optimization. Here we have two runs that we thought were the best for temperature and time levels of optimization. We had a 70-degree and a 98-degree run. 70-degree was for 20 minutes, 98 for 5 minutes. And in these two images, the before and after, you can see that this is the extraction step that's needed before we do the splitting. You can see that the cell membrane dissolves between these two, which allows the RNA and proteins to actually split between the chips, or else the cell membrane would have been in the way. These size are 50-50 sections, but we hope to perform nanosplits at several sizes, even small as 2020 sections. So what's next for this project? We are going to continue we haven't, from here, we actually haven't done the analysis of abundance of the RNA and protein between the two chips. So what's next for this project is we hope to continue to adjust the temperature and time variables to see what is the most effective for getting the highest level of splitting between the two chips. We want to calculate the proportions of splitting across the different tests. We want to try to get a 50-50 split. And then once we have done the abundance analysis, we hope to use data analysis tools such as FragPipe, R Studio, FastQC, and STAR in order to analyze both protein and RNA in the chips. I would say that there was a lot that I learned. I honestly found it hard to describe because my mind was basically blown wide open for what I thought about science and how performing an experiment actually went. So I learned a lot of problem solving skills. That was a major thing in this lab. They would find an issue or a challenge, and then they'd have to work on how to solve this problem. What can we do? How can we change our protocol in order to get the results that we are looking for? I learned a lot about professional interaction and networking. Working on a national lab meant that there was a lot of team science. There was many different groups and different um, people that you had to network with and communicate with to make sure that we're all on the same page and we're all able to work on the same project. That was a lot of fun to do. And I also got a lot of graduate school advising because since I am a senior, I was at a point in my life where I was looking to see what other people had done in their educational careers to get where they were at PNNL. So I received a lot of great advice from different people. I received a lot of recommendations of which schools to look into. And especially as I was reevaluating what I wanted to do with my future and work. I was loved just talking to people and seeing what they've done and what they're doing now. I would like to give a special thanks to my project team, Dr. Uh, Pasatolik, who is the host, James Fulcher, Kevin Zamantis, who are my lab coordinators. James is actually one of the minds behind the nanospots and nanosplits. And Rashmi Kumar, who is a researcher, she has helped me a lot. She's been basically my number one communication, and she's taught me a lot about how to work in the lab. And I would like to just thank all of them for all the work. And the pictures above are some other interns at PNNL who have I've not worked with. And then the other picture was me in the clean room because we had to actually make the nano potch chips by hand, and that was just really fun. So I put that picture there. So thank you so much. And here's some citations if you want to read up more about nanopots or nanosplits.